Good evening. My name is Howard Edelman. I'm your host for Israel and the Jewish World. In The Paradox of Aging, in the last of our interviews with medical research scientists at IMRC, the Institute for Medical Research, Israel, Canada, Ehud Cohen is my guest. His research focuses on the mechanisms of late onset neuro-degenerative disorders. He works on worms and mice to understand Alzheimer's disease. He has demonstrated that aging can be manipulated relatively easily. Can our lifetimes be extended? Can the symptoms of one of the most dreaded diseases of all, Alzheimer's, be reversed? As Woody Allen says, aging sucks. So why do we prefer our scotch aged? Aging matures the flavor, we are told. Relationships mellow. Has that happened between Israel and Jordan? In any case, isn't the real issue living better, not longer? Two rabbis discuss the matter. Stay with us for a show that does not get staler with age. This is another of our series of conversations on innovations in science, and particularly in the medical field. And it's in conjunction with the Institute for Medical Research, Israel, Canada, and our guest is it's Ehud Cohen,
yes,
right?
That's correct.
Welcome to the show. You're in biochemistry and molecular biology.
You can say so, yeah.
Okay. Now, in your field, you're doing work on aging, but before I get into this, one of the things I've noticed in interviews here is most of the education is done, as you did, all at Hebrew University. You got your first, second, and third degree here, right in your doctorate, and then you go off and do your postdoc in the States or somewhere else. Is that a general pattern in Israel?
It is. I'm not sure that most people stick to one institute and do all 3 degrees in one place, but it's very typical that after completing the PhD, scientists are going abroad for a postdoctoral fellowship in order to be exposed to new techniques, new approaches, and study new developments and new things that they can bring here. So going abroad after the PhD is very typical. Sticking to the same institute for all 3 degrees, I'm not sure, but that's what I've done.
Yeah. But the other factor I've noticed is, in your lab, are there any postdocs in your lab?
Yes. There is one, and there is a second one who's going to join later this year. But it's true that we don't have many postdocs in the Hebrew U compared to American institutes.
That's what I was surprised about. You see, you do your undergraduate training here, your graduate training here, then go abroad for your postdocs. Most people go to several institutions to their PhD, but every institution usually has a significant number of postdocs. So I would think that one of the big things is to get funding for your postdocs here.
This is correct, but I think there is some historical aspects, because I would say maybe a decade or 2 decades ago there was a significant difference between the capabilities of the institutes here and in the United States. But I think that this gap is being closed in the recent years, and maybe in the future people would stay here for postdocs in here as well, rather than going abroad. Still, at the moment, I think that most people who want to develop their career and stay in academia, they're still going abroad.
Well, I think it's great to go abroad, but I think Chinese students and students from all over the world should come here, because you're now at the forefront in many areas of science. But there doesn't seem to be the funding for the postdocs to do that.
That's correct. Funding is one problem, and still, I think, because, as I mentioned before, because of historical reasons, institutes like to hire people who spend time abroad. I think that it should change in the future.
Now, you're doing work on aging, and there's this general formula that I read in your papers, and it's not just your research, but generally, that aging is genetically related. But if somebody reads that, it could mean many things. That it's you're programmed to die or something like that, or you're specifically programmed to die at a specific time, or specific kinds of diseases are going to kill you off, or that kind of thing. What's your general take on that, and what's your innovation in that area?
So first, aging is a very complex process, and it has several aspects. Some of those aspects are stochastic. For example, exposure to chemicals or irradiation in the environment and so on. But there are genetic regulated pathways which regulate the aging process. It was shown about 17 years ago in 1993 for the first time that by hitting specific genes, the lifespan of worms can be extended, doubled, and even tripled.
So if we got heated up, you mean there's a possibility we could live 3 times as long? As old as Abraham?
No, I'm talking about
worms.
First, I'm talking about worms, where the lifespan is way more flexible compared to mammals. But I'm talking about the specific signaling pathway of the insulin IGF hormones. Now, if you reduce the activity of this pathway, you can achieve much longer lifespan in worms. Interestingly, later on it was shown to be true for mice as well. And in the last 2 years, there were papers coming out claiming that in a centenarians population, people who live more than 100 years, there is a high correlation between the longevity and specific mutation in this pathway. So
really?
Apparently the longevity mechanism is conserved from worms to human beings.
So they have a gene that makes them live over 100?
They have actually a mutation which either make one gene work or one gene product less active, or a different one which is downstream working, being more active. And that actually is correlated, at least correlated with longevity at the level of human beings. At the level of mice, you can hit specific genes and extend lifespan.
I see.
Yeah. So you ask about my aspect about the aging. So I'm basically a cell biologist of protein aggregation. That's what I've done during my PhD. And I was interested in why genetic cases of neurodegenerative disorders, such as familial Alzheimer's, familial Parkinson's, onset late in life. Typically, these patients are diagnosed around their 50s or late 40s even sometimes. And there's a very basic and important question is where the disease is for 4 decades. The mutation is there, the proteins which aggregate are there, and still nothing happens for 4 decades. And that's the major observation I came with to the aging field, asking whether the aging process play an active role enabling these disorders to onset late in life.
We'll take a break and we'll come back to continue this discussion about the relationship between aging and Alzheimer's, etc.
Okay, sounds good.
Ehud, we'll get back to aging and Alzheimer's and the connection, but first, a curious thing, because you talked about using worms. And the first thing, well, do worms get Alzheimer's? Why do you use worms? What's the benefit?
Oh, it's a very good question, but worms are a very well-established biological research system nowadays, and there are several reasons. First, when you want to study aging, you'll better have a short-lived animal if you don't want to age before your subject.
You get your results.
Yeah, exactly. And worms, on average, they live only for 19 days. I'm talking about small nematodes called C. elegans.
Except if you give them heat, they live for 60 days.
No, give them heat. You can extend it a little bit, but by stressing them, but it's a different issue. These worms have several advantages. Some of them scientific and some others are practical. So I mentioned the practical issue of having short lifespan, but scientifically, we can manipulate their aging process very, very easily using technique which is called RNA interference. And by just feeding them bacteria, and that's their food, they eat bacteria, which express some genetic constructs, we can manipulate the aging process. It's much, much easier than manipulating aging of mammals. This is one thing. Another thing is that these animals have very simple pathways. So unlike mammals, the insulin IGF signaling pathway in worms is only one. It's a merged pathway. And the downstream components are much simpler. Instead of having 4, we have only 1. So it's a great system for searching, for screening, and for studying the very basic fundamental mechanisms. However, it has to be said that at a certain level, you need to validate the findings and you need to test the relevance for mammals. And that's what I actually have done. I worked in worms first, reached a certain level of knowledge which can be achieved in worms, and then moved on to the mice.
To validate what you found in the worms.
I would say to validate and elaborate on, because after finding the specific pathways, we wanted to test first that it works the same way in mice. But mice enables more comprehensive research. For instance, if we're talking about Alzheimer's disease, it's very important to test the cognitive functions such as memory, orientation, which are very difficult or maybe even impossible to test in the worm. But there are well-established assays for memory impairments and cognitive functions in the mouse. So we validated and elaborated on the mouse.
Okay, now when I read a few of your papers, and we'll talk about the specific papers a little later, I formed an image, and I want to know if it's accurate. The image was, the real problem is this garbage collectors are on strike. They're not picking up all these leftover proteins and they're piling up. Or else there's no compactor to reduce them in size. That's the image I formed. It wasn't, I don't blame you for that. Is that any accuracy to that?
I would say that in general this is correct, and maybe I'll say a few words about the mechanistic problem which leads to the development of these disorders.
Okay.
So cells encounter challenges from all types constantly. One of these challenges is protein aggregation. And when protein is produced, it has to undergo processing in order to become functional and to attain three-dimensional structure and to actually mature properly. However, a certain level of, or certain fraction of newly synthesized protein fail to attain this desired three-dimensional structure and expose hydrophobic domains. Now, hydrophobic domains you can, I would say, describe as sticky domains. It's very similar to the little oil that you have on your soup. And you know that those little drops, they don't like to stay apart. They want to create one large drop. That's exactly what proteins do. And we call it protein aggregation. Now, in some cases, protein aggregation leads to disease. Now, what size of droplets are the most toxic? So it has been suggested by others and validated by our studies and others as well, that small, what we call oligomers, small aggregates are most toxic. So cells develop two kinds or two methods in order to get rid of those toxic species. One is to disaggregate them and degrade them, which is the best way to solve the problem because practically it's gone. However, when this pathway is overtaxed or it doesn't function very well, there is a second line of defense, which is compaction or higher aggregation.
I see, so it's sequential, you do.
So we believe that this is sequential and that the first priority, the preferred pathway, is to recycle the amino acids, to degrade the bad proteins, which would solve the problem permanently. However, if it doesn't work well, and we believe that the aging program, the genetic aging program, actually mediate decline in this activity, there is a second line of defense.
It's a backup defense.
Yeah, what we call the compactor. So, in general, that's correct.
I'm glad. I got good marks. But you also have the reason for that. You talk about chaperones. What are chaperones and how does it relate to this mechanism?
So chaperones are proteins which assist other proteins to fold properly and take care of those who did not fold properly. These are actually sets of proteins which serve as, we call it sometimes housekeeping genes or proteins which are responsible for the integrity of other proteins.
Well, I had an image there too of telling your kids, you see, if a protein does it properly, you've got to do the same job when you fold your clothes and put them away.
That's correct.
It's a similar operation.
It's very similar. But of course, they have several roles, not only to assist proteins to fold properly, but also take care of those who did not fold properly. And if their activity is impaired or negatively regulated by aging, problem is around the corner.
I see. Now how does that connect up then with Alzheimer's?
So Alzheimer's is one disease in a group of neurodegenerative disorders that collectively, and actually there are several others which are not neurodegenerative disorders, called
Parkinson's?
conformational diseases. Parkinson's and Huntington's are part of the same group. Now in Alzheimer's, there is a small peptide called A-beta, and A-beta is very aggregative and it tends to form aggregates. Now when these aggregates accumulate, they cause damage to certain substructures in the brain, a process which eventually leads to disease, to the onset of Alzheimer's disease.
But as I read it, it's not, this is the plaque that's formed, is that the plaque? Or
so plaques are actually places where fibrils, which are high molecular weight aggregates, accumulate in an extracellular localization. However, it's not necessarily, in other diseases, it's not necessarily an extracellular localization. Some other diseases, it's in intracellular. There's a whole variety of deposition sites of large aggregates. However, it was shown for several diseases that the most toxic species are the oligomers, are the small and not the large aggregates.
I see. So that's similar to your basic research, in the same that you have to get just a proper amount. You can't have it too little or you can't have it too great.
Yeah, that's correct. I mean, it's apparent from our work. It's suggested and shown by others as well in different aspects and different disorders.
And what I want to do is explore this a bit further, not from the science point of view, the basic science, but how it gets applied to diagnosis and treatment. But we're going to take a break and then come back to that issue. If you can stay with us, don't leave. Ehud, I want to come back to the diagnosis and treatment issues. But first, there's two kinds of Alzheimer's, as I understand it, right? There's what you call sporadic, and then the one that's familial.
I would say it's two patterns of emergence.
Oh, that's what you call it.
The disease is the dynamics is pretty much the same.
I see. So it's the same disease, but two patterns by which it emerges. Okay.
And it differs, of course, in the age of onset.
Yeah, it's what, 50s for the familial?
Most cases, there are some exceptions, but most cases for familial are around the 5th decade of life, while sporadic disease normally onsets at the 7th decade or later.
I see. So, but the mechanism you say is the same, so I presume the diagnosis would be the same and the treatment would be the same, or is that not true?
So regarding the mechanism, it's very hard to tell because there are many unknowns regarding what causes Alzheimer's, and practically we are not sure that there is only one mechanism. However, the dynamics, how the disease onsets, how it's diagnosed, and how it proceeds is well defined, and it's pretty much the same, familial and sporadic.
So if you know that, can you use that to come and tell the chances of somebody really developing if you're 40 years old and you do some tests, or is it that far into the future?
So there are scientific issues as well as legal issues.
Right.
So if we are talking about the familial cases, we know many mutations which are linked to the disease, and they are carried by individuals, members of specific cohorts or specific families.
Right.
Now, practically, scientifically, it's possible to diagnose those people early on and see whether they will develop the disease or not. There are several legal issues which I'm not expert in, and I know that there are limitations about doing that because Alzheimer's, as we all know, unfortunately, is uncurable at the moment.
So it affects their ability to get insurance and jobs and all those kinds of things.
Yeah. And I don't want to
No, that's a separate
elaborate
issue.
on that
Yeah.
because I'm not an expert. It's totally
Right.
separate issue.
But that's only about 10% of Alzheimer's, right?
Up to 10%,
Yeah,
depends
right.
on the population,
Yeah,
but
right.
that's the minority. Majority of cases onset sporadically. And although it's onset sporadically, there are several genes which are known to, and certain variants of specific genes are known to be risk factors. And it doesn't mean that someone who carries this variant would develop the disease, but statistically, this person has higher chances to.
Right.
But still, since it's an uncurable disease, we get to the same point of legal and the social issues which are beyond my expertise, and I'm not the one to ask about it.
But once you know the mechanism, or one of the possible mechanisms, because you don't know how many mechanisms are at work, but you know this is a critical mechanism, how can you connect that up with treatment or some way of dealing with? Because most people I know, including myself at my age, it's one disease that really scares you.
Mm-hmm.
I mean, you can go lots of ways, but you don't want to go that way.
I'll answer this question in two levels.
Okay.
First, in the scientific research level, what we suggest is to manipulate the aging process and prevent disease. Although we don't really understand what's going on downstream, but we do know, based on our studies, that if we slow the aging process, we can prevent the onset of disease. Whatever the mechanism is, it works.
So we gotta exercise, you mean? Yeah.
Now, that's the second level. And here again, as a basic scientist who's interested in molecular mechanisms, human beings are too complicated to understand at this level.
But if you make the worm exercise, do they live longer?
I don't know about the worms, but it was published by others that if you make mice exercise, so it's not that they live longer, but they delay the onset of disease.
I see.
And I think it's widely accepted that exercise, healthy diet, maintaining healthy weight, being cognitively active is a good approach in order to prevent or if not prevent, at least delay the onset of disease.
I see.
Yeah, but it's beyond my expertise, which are molecular mechanisms.
I know, but
Yeah.
the hope is placed on you guys to say, once you get the mechanism straight, then we can figure out how to do it. I know it's a lot of pressure, and you say, well, look, that's not beyond me because it's too complicated. It's in the future, science fiction still. And I've got a big enough job here. I want to get to, because two of your papers became very important. One just in 2008, one 2006 when I was reading the literature, and they're referred to often. Are they they're specifically about this mechanism that you identify, right?
Yeah, so the 2006 paper, which was published in the Science magazine, was addressing directly the question of, is there a mechanistic link between aging and the onset of toxic protein aggregation? This project was conducted exclusively in worms. So this is not Alzheimer's, but Alzheimer-like toxic protein aggregation, I would say. And then we defined, in this paper, we described a mechanism that we defined that we talked about before. The one of them is the disaggregation, which enables degradation, and the second one is the hyperaggregation, which reduces toxicity. Now, I think that the most important thing that we show there, that both mechanisms are negatively regulated by the aging process. So by reducing the rate of aging, by reducing the rate of IGF insulin signaling, we could prevent the onset of disease in worms. And I think that's where the excitement came from. The second paper, which was published 2009 in Cell magazine, we took it to the next level, which is the mouse, and we wanted to elaborate on what we found in the worms. So first, we were able to show that the same mechanism is conserved from worms to mice, and we showed that it's not only protect the animal from toxic protein aggregation, but it actually protects from cognitive decline associated with Alzheimer's disease. And that's the paper of 2009.
Because a lot of references. And you got a prize for that paper, didn't you?
I got a.
Or is it for both papers? I'm not sure.
It's not directly related to the papers, but I won recently a prize from the American Federation for Aging Research for achievements in Alzheimer research.
Now, one of the things, when you get these prizes, they give you recognition, but then they bring also all kinds of attention to yourself, which isn't about the science. I want to know, just as a side issue, how do you handle this? Because you recently were in Canada, you do this public relations, you do the, part of the way you get money for your labs, so you can continue doing your research and maybe for postdocs, which I think helps. How do you feel about that kind of extra work that's added on to your responsibilities as scientists?
We are not in a vacuum. I think being a scientist and working in the academia has several aspects. One of them is to raise money, of course, and to convince people that what we are doing is important. And I think that it's right, because after all, we are spending public money. And the public has the right to understand what he's paying for. Why is it important? And I think that in one hand, bringing the attention of the general public of what is going on in the labs and how it's going to benefit the public in the future is very important. And on the other hand, being part of the faculty means that I have to take part in the general effort to bring money in and to maintain the high level of teaching in our faculty, so on and so on. So I think, from my point of view, it's a package deal. You want to be a scientist, you have to contribute on other aspects as well.
But you know yourself that some people aren't so good on doing that other aspect. They're scientists, but they can't be the public relations. One of the things I've learned, and this is the last thing I'll say, is in these series of interviews, is that Hebrew University professors are not only good at science, the ones I've talked to, they may be pre-selected, so it's not an objective account. We're very good in communicating, and you were excellent, and I want to thank you very much for coming on the show.
Thank you for the compliment, and it was my pleasure being at the show.
My guest is Barry Rubin, and today we're going to discuss Jordan. Welcome.
Thank you.
Now, Jordan is in a particular interesting situation. But before we get to Jordan, I have a puzzle from the conversations we've had, and I want to explore it. On one hand, you appear as a traditional, strong Morgenthau realist. It's power and interests and variations of power in the relationships between states that determine things. On the other hand, I hear you're interpreting domestic politics in terms of agents and structures and ideologies and the war between those. And then sometimes I hear you as it's almost a dialectician, saying the conflict is really of understanding policy is the tension between those internal domestic factors and these external realist factors. Where are you? Can you tell me? I just happen to be a theorist. So
first I'll just say all of those things, but then I can explain it very simply and clearly. I think that one should be a realist and look at interests, but not just interest of countries, but interest of specific regimes. But you have to then understand the specific situation and how they define their interests. In other words, every country is different, and you can't take some system designed to apply to Canada and apply it to Iran. You have to use the same tools, but deal with the specific material you have. So you have to take into account the demography, the terrain, the history, the political culture, ideologies. And then, out of those materials, maybe because of my historical training, out of those materials, you then use your tools. So, to pick a specific example.
Well, use Jordan.
Right. Well, Jordan is not as interesting as some others in this, but
we're on Jordan
it's
today.
simple.
So
it's simple. The key to Jordan is its location, its size, its relatively low population, its relatively low wealth, and the division of its and having to deal with larger surrounding states, and the composition of its population. Jordan is made of two groups: the East Bankers and the Palestinians. And the political power is held by the East Bankers, and they want to continue to hold that political power. So since Jordan has historically been threatened by Syria and Iraq, especially, and historically Egypt, that it needs to counterbalance those threats. That's why it's been so close to England. That's why it's been more recently so close to southern North America, the United States. They have to play at being very antagonistic toward Israel, but they understand that Israel is less of a threat to them than other Arab states. But in domestic terms, because of that Palestinian population, they have to make a good game of showing how militant they are. And if we had the time, we go through the history. But I'll tell you something interesting. I wrote a book called The Arab States and the Palestine Conflict that covered it from 1920 to 1956. And one of the things I noticed is that with Egypt, Jordan, Iraq, and Syria, and Saudi Arabia, there was a certain pattern, and under very different governments in some ways, their basic posture to the conflict in the 1920s and 30s really followed that up to today. That Egypt and Jordan were always less militant and radical and extreme than were Syria and Iraq. And I explain in the book, why that's so. So what are the issues? So what are the issues from the point of view of Jordan? One issue is that they're very worried about a Palestinian state, because would a Palestinian state appeal to their own Palestinians, who are a little more than half of the population? They're very worried about Islamism because their main opposition is the Islamic Action Front, which is a Muslim Brotherhood organization, and has been increasingly drawing Palestinian support. And maybe the best way to continue with this is to tell you three first-hand anecdotes. The first of these is that I was in Amman some years ago, and I was sitting next to a Palestinian Jordanian who was a former ambassador, and across from me was a newspaper editor who was an East Banker. The East Banker said the Palestinians aren't really loyal to the king. The Palestinians said, "Oh, that's ridiculous, that's not true." So the East Banker said, "Okay, quick, tell me, who do you support more, King Hussein or Yasser Arafat?" And the guy, there was silence. And the East Banker said, "You've proven my point." So this knowledge that half the population might not be loyal to them. The second anecdote is that I was sitting next to an extremely large and tough-looking Jordanian general and a Palestinian, I don't want to call him an intellectual, but basically a Fatah hack. And
very different than an intellectual.
Well, you have to know this specific person. And so the Palestinian guy says, "You betrayed us because you made peace with Israel." And the Jordanian general, who's very close to the monarch, drew himself up and said, "How dare you say that? You made the Oslo deal. And once you made that deal, okay, you made a deal, so you negotiate. We can go and follow our own interests." And I think that's an indicative story. And the third story, which I've never told before, is that...
First time on television.
First time ever, actually, I think, is that just before the Kuwait War, so it must have been very late 1990, I went to Amman and met with a group of Jordanian military officers, and they were talking about the Iraqi invasion of Kuwait. So someone brought up the question of, well, what are you really worried about? What do you do? And they said, what we have to do is we have to make sure that Iraqi forces don't invade Jordan. And one of the officers said, not knowing that I hurt him, we'll fight them and we'll get the help of anyone we need. So these kinds of experiences, so if you say to me...
Let me just ask that question. In that last remark...
And now they're worried about Iran.
Was that a reference back to how Israel helped Jordan in Black September in 1970?
You betcha.
It was a directive virtually...
Well, I mean, that's the way I interpret it. So now they're afraid of Iran, they're afraid of Islamists. So if you say to me, okay, all the Jordanians are concerned about is the Israel-Palestinian conflict, that's all that's important to them, and they're just antagonistic to Israel. I mean, this is not what happens, in the real world.
At Jordan now, though, I got the sense that Jordan is afraid of a Palestinian state from you. I mean, that's your...
Well,
but they would support a deal, wouldn't they?
Well, yeah. If somebody else made a deal, they'd have to support it. But that doesn't mean that they're saying behind the scenes to do it. They're also, by the way, of course, afraid of a big conflict in which Palestinians from the West Bank might come into Jordan because they don't want any more. So that's also... They have no interest in going back. It's like they got rid of it. They don't want it to come back because that would give even more Palestinians. So they're very insecure. They have a lot of problems. It's a miracle that the country has survived over the last 50 years. But in the end, they actually managed to survive pretty well. And it's not they have elections, but they fix the elections to make sure the Islamists don't win. I mean, they use repression, but compared to Syria, Iraq, they're much milder. So they've actually managed to work out an approach that works very much for itself. But again, you can't say the entire Middle East is all about the Arab-Israeli conflict. That's all that's important. That's a central issue. It's not. It's just as if you couldn't reduce Canadian or European politics to one issue. From far away, less. When you get close up, it's totally different.
I want to thank you, Barry, again. And as usual, you're very enlightening on these things. And I wish we had more time for all your anecdotes because they really enrich the theory. Thanks.
Thank you.